Welcome, everybody. It's my pleasure today to introduce Sarah McGuire, who will be telling us about classification of Euler characteristic transforms using convolutional neural networks. Sarah?
Thank you. Thanks for the introduction and the opportunity to speak here today. As was previously said, I'm happy to be interrupted with questions as I go. So the overall goal of this work is to be able to summarize the shape of leaves and to use convolutional neural networks to compare the summary representations. We'd like to be able to do this in a way that does not require that the data is pre-aligned, which can be computationally challenging, and in some cases, it's just not really possible. You can see here some examples of leaves that were scanned on the left and some vein outline representations of them on the right. And there's a natural way that all of these leaves have been lined up because there's generally a stem that they grow from, and you can orient them all in the same way, computationally, of course. But there are some instances when there's no canonical way to orient your data. For example, if you have 3D point clouds representing protein structure, proteins are all different sizes and shapes, and there's no long and short way, so there's no natural way to align them. So our goal here is to have a TDA-based classification method, which does not require this alignment. So I'll start by reviewing the Euler characteristic transform as a method to summarize the shape of data. Then I'm going to talk about the convolutional neural networks and why they make sense to use in this context, along with some nice properties that we get from them. And I'll then describe the specific ECT-CNN framework we're using and show two different applications to leaf-shaped data sets that come from our collaborator Dan Chitwood at MSU, who's in the Department of Horticulture and studies plant morphology. And this is all joint work with my PhD advisor, Liz Munch, as well as Dan Chitwood, who provided all of the data and the biological context for us. Okay, so starting with the Euler characteristic transform, you're probably all fairly familiar with the Euler characteristic. It's a very simple summary, one integer that we can compute from a polyhedron with the vertices, the edges, and the faces, just by taking an alternating sum of their count. And we can do this for any space that we're able to triangulate. The Euler characteristic is a topological invariant, so we know that any homotopic spaces will have the same Euler characteristic value. But instead of computing one Euler characteristic value for an entire shape, we instead want to select a direction, omega, define a filtration, and compute the Euler characteristic of sublevel sets of a shape as we move through the filtration. In this case, I'm assuming that the shape is an embedded simple shell complex, so we have coordinates for each of the vertices, and we can define a simplex-wise function in the filtration f omega by using just the standard dot product of the direction vector with the embedding for each vertex. Using this filtration, we can sweep through the simple shell complex in the direction omega and compute the Euler characteristic curve, which is a map from the real numbers to the integers. And here the filtration function values A get mapped to Euler characteristic values of sublevel sets at that filtration value. And we have this nice step-like function thing, which is really just a vector of integers. So what we'd like to do with directional transforms in general is, instead of choosing one direction and computing a topological summary for our filtration in that direction, we'd instead like to compute the topological summary for many different directions and get a collection of functions. So specifically, the Euler characteristic transform, or ECT, is what we get when we have one of these directional transforms. And the specific choice of topological summary we're using is the Euler characteristic curve. The original paper from 2014 is written with a focus on the persistent homology transform, which does the same thing, except instead of computing Euler curves for each direction, you're computing the persistent homology, and you get persistence diagrams for each selected direction. So for the ECT, instead of having just one function now that maps the real numbers to Euler characteristic integers, we instead are taking all the directions and we get these collections of functions called the Euler characteristic transform. And we have a map from the directions on the d-1 sphere to the space of functions from R to Z. So specific directions omega map to functions that map values in the filtration function to the Euler characteristic of the sublevel set at that threshold. So the ECT combines the simplicity of the Euler characteristic as our shape summary with also having many different vantage points. So that makes it a very simple but also powerful tool to represent the shape of our data. This is true in practice because we can actually compute these things, but it also has nice theoretical guarantees, including the fact that it's an injective map. This tells us that it's actually a faithful representation to use for our data and can be used for shape comparison. The ECT injectivity proof for simplicial complexes, specifically embedded in dimension up to 3, using all possible directions, is in the original 2014 Turner paper, and there are also other extensions of this in other papers to just the general space of constructible sets in RD. Also, it was shown that this injectivity holds for only a finite number of directions, which is important for actually computing the ECT of real data. And having this injective map is a very important motivation for using the ECT for shape classification, because given enough directions, then we know that the ECT of two shapes, if the ECT of two shapes is not equal, then the shapes are not the same, given that they're in the same space. One of the interesting variations of the ECT is the smooth Euler characteristic transform, which has some nice theoretical properties. Because the Euler characteristics are integers, if we just use the regular ECT, we end up getting these step function things, which causes some statistical issues in practice and also creates stability issues. So to avoid this, the SECT replaces the Euler characteristic curve with a continuous function. So I have like continuous version and discrete version here. The definitions of the SECT are continuous to ensure that we're actually able to prove things about it, but also I care about how this happens in the discrete case, since that's the setting I'm dealing with applications that I'll show later. I should say that we're also assuming we have finite embedded simplicial complexes that are contained in a bounding ball of radius t. So for the computation of the ECT in our specific data sets, what we did was we found a global bounding ball for the entire data set so that we can have the same number of thresholds of a fixed size for each direction of each sample in the data set. So we don't have differently sized thresholds for different directions. So the result is we have this bounding box from negative t to t, and I have a set number of thresholds from t0 up to tj. So to get from the ECT to the SECT, we first have to have the ECT centered. So what we do is we compute the mean value of each Euler characteristic curve, which is just a vector. So you can just compute the average of this vector. Then zero center each of the Euler characteristic curves using this mean, which is shown in the middle step, which is the same for the continuous or discrete version. And then finally, we get the smooth Euler characteristic curve by integrating over all of the function values in the filtration. So equivalently in the discrete case, we're just taking a cumulative sum over the thresholds in our filtration. So beyond the applications on simple test data for variations of the ECT that are in those respective papers, there's been other efforts to actually use the ECT for analysis of real data and using it in combination with some sort of machine learning capacity. So in this first paper on the left, Eric Amesquita and his co-authors used the ECT to quantify the shape of barley seeds. What they do is they can concatenate all of the ECT vectors into a high dimensional vector and then perform dimensionality reduction on it and then pass it into an SVM for classification. Because they're using an SVM classifier, this method requires that the data be pre-aligned. And because they're concatenating the ECT into one long vector, we're losing and ignoring information about which direction each of the Euler characteristic curves came from. In this second paper, they define a weighted version of the ECT that uses a simplicial complex and an associated weight function as the input to the model. And they motivate this by the existence of brain tumor data analysis, because I guess this data often comes in the form of segmented grayscale images, which they could represent as a weighted simplicial complex like this. So when they're applying the weighted ECT to a classification task for the MNIST handwritten digits, you can see an example here of the digit 2. They also use an SVM model to classify the ECT data, which similarly necessitates the alignment of the data and means that the directional information is not being used in the classification model step. So in this third model, or third paper on the bottom, they're not using the ECT as a static shape descriptor, like the previous two. The ECT data is not the input to the model. Instead, they're using the ECT to define a layer within a neural network. And they say that this can also be used as a loss term. But in order for the ECT layer to be updated using backpropagation, it must be differentiable. And so they modify the ECT to define a differentiable version, which ends up being differential specifically with respect to the number or the directions chosen when you're computing the ECT and the coordinates of the input data. So now I'll switch gears a little bit and talk about convolutional neural networks. I use CNNs throughout to refer to them, but ConvNet, it means the same thing, if that's the term you are more familiar with. Just some background context of why this is the right tool to use for ECT data. So there's different types of machine learning models that are better suited for different types of data. For any type of data that lives on grid-like structures, for example, images, a grid graph, even time series, if the grid is just like 1D, CNNs are designed to leverage that structure to come up with the low dimensional embeddings and then represent them and use that embedding for classification. They use the regular grid pattern to gather information about local neighborhoods. So if your data is an image, like the image of a dog here, you could just take each of the columns of pixels as a vector and concatenate them, and you have one long flattened vector. But this probably isn't the best way to represent an image, because now we're losing structural information that contains important context. Now, obviously, these squares are exaggerated as pixels, and they contain more than just one pixel. But as a human, we can look at this vectorized version on the right and maybe like piece together the jigsaw puzzle and determine that this represents a dog. But for a model like an SVM or a multi-layer perceptron, determining that this vector of pixels is a dog is a bigger challenge because we've lost all of the contextual information. So CNNs are optimized to actually leverage this contextual information, specifically by looking at local neighborhoods, and the information can then be extracted from each pixel's relative location to other pixels. So the basic building block of CNNs is the convolution operator, which is how we define convolutional layers in the neural network. And very generally, convolution is an operation on two functions, x and h, that produces a third, x convolved with h. To define it in the continuous setting, you first reflect one of the functions over the y-axis and then shift it. And then you integrate the product of these two functions. In this functional setting, it is measuring how the shape of one function is modified by the other. So in that sense, it's sort of similar to a cross correlation. But we'd like to think about CNNs as operating on images just from the same signal processing perspective, even though we're talking about images now instead of functions. We can think of X as our image, which is just a signal on an n by n grid. And we also have another signal H on a smaller grid, and we call this our filter or our kernel. And we'd like to convolve the filter with our image. And so we compute X convolved with H as the sum of the product of the two signals with the same reflection and shift. And here U and V denote pixels. So the sum over V can be restricted to just the support of the filter in this small neighborhood of U. So V are the immediate pixels surrounding U, the pixel U. And you can see what this looks like on this simple toy example on the right. We have a grayscale image with pixel values shown on each pixel. The pixel values are shown on each pixel. And we have a small filter shown in pink. And to convolve the filter with the image, we center the filter at a pixel value, take entry-wise products of the pixel values, add them together, and that single number is the result for that pixel location. Then the filter just moves to the next set of pixels. We recompute our product and sum, and that's added to the output. We call the output values here the feature map, and there's many different choices of how you can, the size of this feature map. There's parameters that you can change that would affect this, but for now, this is just a basic example. But in CNNs in general, these learned pink filters, these are the parameters that the model is learning. These are the values that get updated through training iterations of the model. So now I'll define two different but very generic properties of functions that are very important when we talk about the behavior of neural networks. The first is equivariance. So if we have some transformation to our input data, so it's a function that would be equivariant with respect to that transformation satisfies this statement. Essentially, if we pass our input data x, which has been transformed by t into our function, we get the same result as if x was passed into f without any transformation, and then the transformation was applied. So the output is transformed by an equivalent amount, hence the name equivariant. And in this example below, if we think of our CNN model as approximating a function which takes in images and the output is where in the image is the animal sitting, then the first image we see is a dog sitting in grass, so the model classifies this as grass. Then if we transform the image, in this case, the dog is translated across the image, then the output is equivalently translated to be sand. So this is just a toy example of this. And the second property I want to talk about is invariance. In this case, we say that a function f is invariant with respect to a transformation if transforming the input before passing it to the function does not change the output at all. In this example, I have another image which is classified as dog, and if we apply a similar transformation, just translating the dog within the image, the model still classifies this as a dog, and so it's an invariant function. As you can imagine, this seems like sort of an obvious property that we'd want an image classifier model to have for most tasks. However, actually encoding these properties into CNN architectures is not always obvious for different types of transformations. And there's a lot of active research in invariance and equivariance properties of specific deep learning architectures for different classes of transformations like rotation, scaling, translations, and so on. But one of the important takeaways here, and the one that I'll focus on today, specifically for convolutional neural networks, is that they are translation equivariant. We know from the functional analysis definitions of convolution that the operation commutes with respect to translation. So the convolution is defined by reflecting and shifting one of the functions before you take an integral. And so convolution as an operation is commutative because the choice of which function you reflect and shift does not affect the integral result. So that means that our convolutional layers within a CNN are translation equivariant. In order to get to translation invariance, meaning that all the translations within input images don't affect the output classification result, there's a bit of a gap between what we can theoretically prove versus what is found to be the case in practice. So there's three common ways that invariance is encoded in deep learning architectures. The first is through pooling layers to sort of average features that are extracted, also through data augmentation, and generally just having large, diverse data sets. And finally, by just hard coding these equivariant architectures. For example, what we see for the convolutional layers, which we know are equivariant. So overall in practice, the deep learning community seems to be on board that translation invariance can be achieved, so long as your data set is large enough and diverse enough, and through the use of this data augmentation, which essentially just means you're applying transformations to your data set to get more training data. And there's this really interesting paper by Bischioni and Bowers called CNNs are not invariant to translation, but they can learn to be, which tackles this problem. It's sort of an interesting perspective. So now I will describe the specific ECT-CNN framework that we are going to work with and what we're proposing. This is an overview of what we'd like to do with the ECT and CNNs. If we have an input data that's some kind of embedded simplicial complex, we compute the Euler characteristic transform. And then what has been done in the past is just concatenating the vectors and passing it into an SVM classifier. Instead, what we'd like to do is represent the ECT as an image and pass it into the CNN to classify. So the reason that we're able to represent the ECT as an image in a way that makes sense is because 2D ECT data lives on a cylinder. And so we can take our input simplicial complex, we can pick a direction on S1, compute the Euler characteristic curve. It's just a vector, so we stack it up. And then if we compute these Euler characteristic curves all the way around S1 and stack them up, we get this cylinder. And then to represent our cylinder as an image, we just pick a place on the circle to slice and flatten it down, and we get a normal 2D matrix. So this matrix has directions sampled from S1 on the x-axis, and the columns are just the Euler characteristic curves. So we obviously can't store a data as a cylinder in a way that makes sense. So we represent the ECT as this 2D matrix. What you can see here are two different color patches of ECT values on the cylinder, a green and an orange patch. And if you think about the patches rotating around the surface of the cylinder, that would correspond to translation in the flat matrix version. And also on the edges of this sort of arbitrary seam we picked to cut and to make the flattened version, we should have copies of either side appended to it to give the illusion of the edges are actually the edges being identified. So the important point here is that because rotation on the cylinder is the same as translation in the flat matrix of the ECT, we're able to rotate the input shape, and the effect is translation in the ECT image on the right. We're able to prove that for finite embedded simplicial complexes in R2, that rotations of K are equivalent to translations of the ECT, which we can represent as translations in the matrix form. And then since our CNNs are translation equivariant, that means that our ECT-CNN framework should be rotation equivariant. So in order to give the CNN the illusion that the image lives on the cylinder, we use padding defined by built-in methods as part of the PyTorch package. So for the sides, what we want are the left edge to be identified with the right edge of the image. We use circular padding, which just uses copies of columns from the right side to pad the left and copies of the left side to pad the right. You can see this in the middle image that shows circular padding just from PyTorch. On the top and the bottom, we're just choosing to pad with 0-valued pixels, which is a standard choice for normal images in CNNs. And the choice of how much padding to use, and how many pixels deep we'd like to pad our image, that's sort of a user-defined choice, but there are a couple parameters that you can mess with that would have effects here on the output feature map size. So one of the standard choices here that you can define is called stride, which is a parameter that controls how far the kernel is jumping every time it's shifted. So when S equals 1, the kernel or the filter stops at every single pixel and is centered there for convolution. But when the stride is greater than 1, we have the effect of the feature map shrinking because we're not stopping at every single pixel for convolution. And that can be good for dimension reduction and reducing the computation time because the filter is actually applied less times. But it also has a trade-off and can cause the model to lose information, obviously, especially more fine-grained details depending on what the images look like. But if we assume that K is our kernel size and we assume that the stride is 1, if we define the padding to be K divided by 2, this has the effect of preserving the feature map size. So that means that the input image size and the feature map size would be the same, which is what we choose to do for our implementations. So a major aspect of this analysis pipeline relies on the fact that we don't have to pre-align our data. So instead of just assuming that the data is unaligned, we actually introduce random rotations of each sample to ensure that it's not. This is sort of often a challenge in deep learning. It's common to have a model which is overfitting to an unintended signal in the data instead of this actual signal of interest. So for example, a CNN classifying images of different modes of transportation might only be extracting features from the image backgrounds and ignoring the actual vehicles themselves to classify the images. So that's the behavior we saw in both leaf data sets that we applied this to. If you don't introduce these random rotations, the CNN models actually show higher classification accuracy values than when we apply the random rotations first to get rid of any incidental alignment. And that's indicating that the model was overfitting to the orientation of some of the leaves, as opposed to just the signal we care about, which is the shape of the data that's measured by the ECT. So this is an overview of the CNN architecture we're using to classify the images. We're using grayscale image inputs, so it's a single channel and it's a 32 by 48 image because we're using 32 directions sampled on S1 and 48 equally sized thresholds. We use 2 convolutional layers, each with max pooling layers paired with them, and flatten the representation and pass it through to 2 fully connected layers at the end. So most of the architecture parameter choices here were not done in a specific way, just they were selected somewhat randomly. Like for example, the number of channels in each convolution layer. That's because we're not really trying to get state-of-the-art results that are competitive with models trained on ImageNet or anything like that. What we're trying to do is just evaluate the method and the utility of analyzing ECT data using CNNs. So I'll now switch gears a little bit and talk about the application of this method to 2 specific data sets. The first is a leaf graph data set. This has 40 different classes of passiflora leaves, and the passiflora leaves have a lot of variation within different species. So our 40 classes represent different species. We're going to represent these as embedded graphs. They all have 15 vertices and 24 edges, and there's only about 100 samples for each class for this data set. Our second data set is much larger and gives us much more refined boundaries for the shape of leaves. So we have only 14 classes of general plant families, and the size of these classes, the number of samples we have for each class, ranges from about 1,000 to over 80,000 samples. And we also represent this data set as embedded graphs. So this first data set, the passiflora data set, has about 3,000 leaves from 40 different passiflora species. And in the paper where they released this data set, they define landmark points, and all of these graphs, or all of the leaves, are pre-aligned. They're rescaled and pre-aligned. What we do is we represent them as embedded graphs by taking each of the 15 landmark points that are existing in this data set and their coordinates to represent them as graphs. So each landmark point becomes a vertex, and we connect all of these 15 vertices using the same 24 edges. So all of the graph representations of these leaves have the same adjacency information. This is just a choice we made based on this data set. You can see an example here of what the embedded graphs actually look like for 3 slightly different examples, but we're able to capture some of the vein structure that exists in these landmark points on the leaves. But all of the input graphs to this data set have the same adjacency structure. So they have the same number of vertices and edges and the same edge connectivity. You can see an example here of if we're computing the Euler characteristic curve in one direction, we get this vector here. And if we compute it in many different directions, specifically 32, you get the ECT image versions here for these 5 different example leaves that come from different passiflora species. I also have the SECT versions represented below, and these are just some examples so you can see what they look like. Obviously, they're much smoother images, which is the point of the SECT. But we'll see in a second that classification-wise, we don't necessarily find that this method, that using the ECT would improve on our method. So in terms of classification results, what we did to compare different models, our model is this ECT-CNN in the middle. We have classification results done using tenfold cross-validation with the average values reported here. We also, to compare them, we also wanted to use just the flattened vector version and pass this into an SVM, which is this result here. Obviously, this classification accuracy result is very bad. We're using unaligned data. We had introduced random rotations to all of our data set. So it makes sense that the SVM performs very poorly here. As another form of comparison, we also just took a low resolution image of each leaf graph and passed this into a traditional CNN for classification. And we chose the specific resolution of these images to be 32 by 48 so that it would be the same exact resolution as our ECT images, just for consistency of comparison. So we also take our ECT and we compute the SECT from it and pass that into our CNN pipeline. This also performs very poorly, which is sort of an interesting result, but what we found is that this is largely a result of us keeping all of our parameter choices the same across our different models here. We kept the model architecture exactly the same, so we have the same number of learnable parameters, same kernel filter sizes for everything, just for consistency of comparison. But if you increase the kernel size for convolution, when you apply this SET-CNN, you can boost the accuracy up to match the ECT, which makes sense because the SECT encodes exactly the same information that the ECT does. It's just the smooth representation of it, but you can move back and forth between these two. And so finally, what we did last to compare, which has slightly outperforms our ECT-CNN method alone, is that we just take, instead of a single grayscale image, we take a 3-channel image now with 2 copies of the ECT and 1 copy of just the graph image itself stacked together, and we pass that into a CNN. And this method performs the best. So it's the TDA method combined with traditional method. The second data set we consider is much, much larger. It is over 162,000 leaf outlines and represents a very wide variety of plant groups of different flowering plants. We also represent these as embedded graphs. You can see the 14 different plant groups shown here, just some examples of what they look like. And also the distribution of classes here. You'll note that it is a very unbalanced data set, that the tomato class has 82,000 samples, while some of these smaller ones have less than 1,000. So it's a very unbalanced data set to work with. But we do represent these as embedded graphs the same way as we did for the other ones, relying on the fact that we have leaf outlines and we know the order of all of these points. We can build a graph. These are much, much larger graphs. I've zoomed in here. You can see that it has many, many vertices and edges that make up this leaf outline. So similarly, a few examples of what the ECT and SECT look like for 5 different classes. You'll note also that we have sort of this dark space here where there's just 0 valued ECT, because that's a result of our bounding box for our entire data set. So in this data set, there are a few leaves that are much larger. And so our global bounding box for our data set means that a lot of the leaves have this sort of empty space here because the bounding box is very large around the leaf. The SECT looks a little different here, and you'll see there's not so much, it's still smooth, but the sort of smoothness is less global. So we have less of an effect when we get to the classification accuracy of the filter size on accuracy. So as I said, going from our regular ECT-CNN to the SCCT, our performance doesn't drop as much. And I would say that's primarily because of the size of the data set, that we still have a lot of data to train on and does very well, but also the scale of structure here isn't as global as it was for the other data set. And so the effect of the kernel size is reduced here. But similarly, we compare against just the flattened vector version and pass it into an SVM. We take just an image version of it and pass it into a regular CNN, which does outperform the regular ECT version. CNNs are optimized to work very well to classify images. So it makes sense that we'd be able to do very well on a data set that's very large and diverse to classify well with just a standard CNN. And finally, again, the best performing method is when we combine the TDA with the traditional methods. And so we're able to boost the performance of our traditional CNN by also including these ECTs stacked in to create a 3-channel image. So in summary, with this method, we're able to summarize the shape of leaves using the Euler Characteristic Transform and represent these summaries as images. And images are very well suited for classification using CNNs. We're able to use more of the directional information in classification as opposed to the SVM methods, which are just using the flattened ECT vectors. So the SVM classification doesn't give us any rotation equivariant properties. And because if you change the orientation of the data and then recompute the ECT and flatten your vector, you've lost all association from one direction to the next. But by using the CNN in this way with cylindrical padding, we're able to get a rotation equivariant pipeline. And so our input data here does not have to be pre-aligned, which can be a computationally expensive task. And it's also really useful in the case that there isn't actually a natural way to align your data. So we see some consistency between these two data sets in which models came out with the best performance, but we do see that the best performing model for both data sets uses the ECT in some way. And so for the graph data set, the standard ECT-CNN and the TDA combined with traditional CNN showed the best performance. And for the larger leaf data set, the leaf outline one, we see that the best overall performance came with the stacked image using both TDA and traditional combined. So there's a lot left to unpack here in terms of interpreting the leaf ECT embeddings, like from a biological perspective. I know our collaborators are particularly excited about being able to use TDA methods like the ECT to pair with existing morphometric analysis techniques that rely on more standard measures of leaf shape. And we'd also like to continue the analysis with more leaf data that they have, but we haven't used yet. There's also work to be done with the translation, equivariance, and invariance properties of this pipeline. There's limitations to consider that we're using just these simple out-of-the-box CNNs like this, and there's potentially ways we can adjust the architecture we're using to hard code the translation invariance into the model, as opposed to right now where we definitely have equivariance, but invariance comes mostly from training considerations. We've also thought a lot about what this pipeline could look like when you're working with 3D data. And while the math is very clear for the ECT of how you jump up a dimension, it's less clear how you can directly generalize this method to 3D because of the CNN. And so for 3D data, instead of the ECT living on a cylinder, where you can represent it as a 2D tensor with the two edges identified, now you have ECT data that lives on a hypercylinder. And so you need to have a way to sample S2 now in a way that you can represent all of the directions in a grid-like way. And there's not really a good way to do that. There's no coherent way to sample S2 that you could map the directions to a grid. And there is a preprint by Tejan Paik that uses 3D ECT data with a neural network. But what they do to circumvent the issue of sampling directions on S2 is they have a graph neural network used as an intermediate step. And so they represent the directions on S2 as a graph as opposed to requiring just the strict grid format, and that circumvents this issue, and then they can pass it into a CNN downstream. And finally, another thing of active work that we're working on is applying this to 3D protein structure, which is ongoing work and obviously requires the extension of this to be able to do it in 3D when the leaf case is much simpler, representing everything as 2D matrices. So I will end with that. Thank you so much for your attention, and I'm happy to answer any questions.
Wonderful. Before we get to questions, let's briefly unmute ourselves and applaud for the speaker. All right. Questions for Sarah?
I would have one. Namely, when you showed the combined ECT with the original image, you said you stacked 2 copies of the ECT with 1 copy of the image. Why 2? Can you explain that a bit?
Yeah, so the reason for that is that working with PyTorch specifically, or I'm assuming with TensorFlow also, but passing it into a CNN, the sort of default is it's expecting an image. So the types of images that these models can work with is a grayscale image, which is 1 channel, or a color image, which is 3 channels, where you have a red channel, a green channel, and a blue channel. And so you really need 1 or 3 in order to just use it directly in this way. And so I just arbitrarily chose to have 2 copies of the ECT and 1 copy of the image, but the classification results don't vary much if you were to do 2 copies of the shape image with 1 copy of the ECT, but you do need 3, just default for PyTorch to just use it directly in the same way.
Thanks, that makes a lot of sense.
Hi, I have a question. Is there any work, or have you thought about how to interpret the ECT output directly? So I can see there are like some curves and some, I don't know if there's something on that, or is always fed into a CNN and then let's see what we get out.
Yeah, I mean, we've visually looked at the images to see just the overall shape of it and how they vary, how consistent they are within classes versus between classes. What we were seeing with the leaf graph data set was that there's a lot of variation within classes and not so much variation between classes. So that made the classification task very difficult, and you can actually visually see this looking at the samples of it. But where there's variation in these images, we looked at like a heat map of where all the variation is, and it's really along the edges of the image. And we measured the variance within class and between classes, and the fact that the between class variance is so low makes the classification task very difficult, whereas for this data set, it's much larger, it's much more diverse. Just by nature, there's less variance within classes than between classes, and so we don't have that same issue of classification, just visually comparing the representations from these from the ECTs. I don't know if that answers your questions. Any other methods other than just visual comparisons?
Yeah, thank you very much.
Sarah, hi.
Hi.
Got a question about the protein data. When you're analyzing the protein data, will you be using the complete structure or just the alpha carbons, or some subset of the complete structure?
I believe the plan is just to use the backbone. Yeah, so like the alpha carbon chains. Yeah. Obviously, there's protein data in general, there's a lot more data to work with. 3D, you have a lot more computational challenges, even just computing the ECT. So that'll be a challenge for trying to directly apply this method just computationally. And I think restricting it to just the backbone helps that a little bit.
And what about different conformations of the protein? Is the idea to be able to recognize different conformations of it, or will they be considered separately?
I'm not sure yet if there's a clear question of what we're trying to answer with this method. I think that comes more from our biology collaborators and what they'd like to get out of this. Right now, it seems that they'd like just a general way to represent protein structures in different families to maybe come up with some sort of phylogeny of how things break out in this like ETC space of of protein structures as a whole, but it's sort of a vague plan right now for the specific questions we'd like to answer about the protein structure.
Good. Thank you. Thank you.
Hi, Sarah.
Hi.
Great talk, first of all.
Thank you.
I'm doing my master's thesis on the ECT right now, so it's been really great to hear what the news are about that. I had a question on this very slide you are here, because I'm seeing that the scale of colors that you're using ranges from -1 to 1. I'm a little confused on how that works, because if I understand it correctly, the ECT just takes like integer values. So I'm wondering how you are translating integer values to values on -1, 1.
Yeah, so the representations I have shown here are normalized. So these are not the raw ECT values and vectors shown. They have been normalized for them to be passed into our CNN model. And specifically in PyTorch, we have these tensors represented from values -1 to 1. So they've just been rescaled.
Okay.
And normalized.
So like value 1 would be like the maximum value the ETC takes, right?
Correct.
Okay, thank you.
Just for normalization for the purposes of our classification model.
Okay, perfect. Thank you.
Further questions? All right. Well, if not, we'll end with the public questions here. So thanks so much, Sarah, for the fantastic talk, and let me end the recording.